Absolutely. Thanks very much. Let me just say what a pleasure. You go on and escort Stefano, but what a pleasure it is to be here. This has been a fabulous meeting. The quality is as good as anything that one can see in grander venues. Although, as I looked about me when I woke this morning, it's almost hard to think of a grander venue or to and I have to express my thanks to Guy and others who made this meeting possible here in Downing, which is really beautiful. Guy stimulated me last night to look at the history of Downing College somewhat, and I was actually extremely pleased because we're all about innovation and in dementia research now, and that's what brings us here. Really trying to break the mold, do something out of the box, and other sorts of metaphors. And we're, oddly enough, in this highly traditional institution, we are in exactly such a place as should remind us that this is what changes the world. So as you go to the art gallery in the front of the college, you'll see a beautiful plaque to the first Baronet Downing, whose fortune was used by the third Baronet. It sounds like people steadily lived on that money for a long time. So the first Baronet's money, which was responsible for these buildings, was made. He was a tremendously entrepreneurial fellow. Member of the first graduating class of Harvard University, but moreover, and this is the really charming part of his history, he was described as one of the greatest rogues in Royalist history because he grew up in Protestant New England, came back and joined the Roundheads as one of their staunch supporters and diplomatic leaders, and then when the tide shifted somewhat in politics in England, he joined the Royalists, was accepted because of his diplomatic connections, and promptly went about leading as a spy the roundup of his erstwhile colleagues, the regicides, and we all know what happened subsequent to that. Anyway, so it's in this spirit of trying to break the mold that I wanted to talk a little bit about neuroimaging and the UK Dementia Research Institute. What I want to do is the topic is a very broad one, and it's one that is a little bit difficult to focus because the UK Dementia Research Institute isn't itself about imaging. And it doesn't have an identified imaging component, but I hope I'll leave you with the impression that it is a collaborative venture extending across many, many people that wishes to grow in its associates and is crying out for solutions to problems that imaging can play a major role in. I've already started to see some of them that have set me thinking over the last day. I'll describe first the overall vision of the UK Dementia Research Institute because I think it's something that is certainly less well understood by some. I'll go into a little bit of detail about the kinds of programs that are in our center at Imperial College, and you'll understand, I'll try to hint in each one of them where I think imaging could be brought to bear with some innovation. I also want to touch on the program in Edinburgh because that's Joanna Wardlow has had to leave, but she is probably the most identifiable imaging-specific representative. But I also wanted to give you a flavor for the diversity between the centers as well as their obvious synergies. And then I'm going to close with a sense of just some hints of areas that I see close to me are emerging where imaging is starting to provide the kinds of novel solutions that we're looking for. And I focused somewhat on MR imaging, given the majority of the audience, with apologies, frankly. And I hope that in the end, what this will leave you with is a sense of enthusiasm for feeling for this venture that has taken off in the UK, and a sense that it's an open exercise, it's not a closed shop. And third, the sense that dementia investigators really are crying out for the kinds of solutions that all of you are in a good position to deliver. Well, at the very start of the exercise to develop a Dementia Research Institute, the then prospective director, Bart de Stroper, before he really took on the job, tried to identify what he really saw as the key challenges for research in Alzheimer's disease. And I want to just highlight these. None of these, I think, are real surprises. And in fact, if you look closely on in some of the previous talks, particularly in Craig Ritchie's talk, I noticed most of these are appearing there. Clearly, we know there's a lot of interest in protein misfolding, and but of course, related to that is the phenomena of disrupted proteostasis, which we hear less about. Mitochondrial dysfunction, which takes us back to the very earliest days of magnetic resonance spectroscopic imaging, has been part of dementia research for some time and still is relatively poorly resolved as a pathogenic issue. We heard about spreading of toxic proteins. James talked to us about dysfunction of the synapse, the action center for the neurons, clearly a hot area for functional imaging, neuroinflammation. And then Joanne gave a tremendous introduction to what is still imaging space, which is the dynamics of both perfusion and disruption of blood-brain barrier integration. And finally, a kind of an emerging area, which, amongst other things, Joanna's recent paper in Science Translational Medicine illustrated, is that there are aspects of homeostasis in the brain that tell us more about the susceptibility to Alzheimer's than probably anything else that we know of to date. Now, what is the DRI? The DRI is a unique sort of collaboration between the two major charities in Alzheimer's disease, Alzheimer's Research UK and the Alzheimer's Society. And Carol Rutledge from ARUK is in the back. She's been interacting with many of you. And the MRC, putting in £250 million over the course of 5 years to seed the development of a virtual center that links a hub at UCL with 5 other centers across the country based in Edinburgh, Cambridge, King's, Imperial, and Cardiff. And these, despite the fact that they are all in separate institutions, the really exciting thing about this, and again, the really groundbreaking thing, is that they're all linked by a set of agreements between the institutions that allow them to function as a single entity. So everything can move freely from one to the other, even down to staff. And all of this is looked after by an independent DRI board, so it has the flexibility as a standalone exercise to do things much more sleekly than usual UKRI types of processes. So I've talked about this being a joint exercise, and the idea of it was to bring together expertise. It was never envisioned that all of the world-leading expertise would be those previously identified or identified prospectively as project leads in the UK DRI. It includes both them and those who they associate with, collaborate with, and otherwise involve in the activities. And the idea is for it to really be grounded in the first instance, for sure, in a drive towards discovery science. It is not about trying to make a drug in 5 years. It is about trying to really step back from the pathology of late life neurodegenerative disease, reassess the processes, and try to find some new starting points to provide a foundation for a much broader set of approaches to their mitigation in the long line, in the longer term, with development of a new therapeutic pipeline over the next 10 to 15 years. The focus for the work is resolutely, and I've really highlighted this, on the initial prodromal phases of the disease. It is not about, in the first instance, about trying to stop the disease once it is fully clinically manifest. It is about trying to intervene, identify, and intervene in the process in the period, decades ideally, before it ever appears. And that is why there's a tremendous amount of interest in the homeostatic and protective mechanisms associated with the disease. I think I'll skip, but the scientific objectives really are the core of what DRI is about. It really is the radical idea, and it sounds bizarre to state it in public, the radical idea is to free investigators to focus on the science and get away from the other sorts of things that we so often need to do in order to survive in an academic ecosystem, by giving them this protective envelope of an overarching organization. But importantly in this, and again, I really, really want to stress this, because this is, you see, in addition to all the things I mentioned before about doing novel things, it's about increasing interactions and crosstalks between research groups, not just within the DRI, but doing precisely what we're doing here today, trying to reach out outside of that limited group of people in centers. And to do this not just with the tools of neurobiology and pathology, but using, trying to bring in new kinds of technologies and approaches. And of course, fundamentally, it's also about profile. So it's about research, it's about communication, and ultimately, this is intended to drive out towards translation, the kinds of knowledge that over a longer term, not over a short term, will make a difference to patients. Now, within the Imperial Centre, we, after some assessment of what we know a little bit about, and I'll be frank with you, I'm not sure that many of us felt ourselves uniquely qualified as dementia investigators. But what we did feel on reflection was that we saw important science that we did know a little bit about, that we could bring to the problem of dementia and try to provide, through that new perspective, some new kinds of solutions. And I think what we really have focused around is the notion that, often forgotten in the enthusiasm for identification of genetic risk factors that may implicate specific molecules, we often forget that it's environment and lifestyle that actually still contribute through directly and through the comorbid diseases that are manifest as a consequence of them, the major risks of this disease. And so the brain, and as neurologists, we have tended until rather recently to forget that the brain does not exist in a vat that we carry on top of our shoulders, but is integrated into a physiological system that we call the body, where it interacts constantly with organs around. And we've heard aspects of many of that. Now, our focus to try to bring this to a tractable set of target entities is really on trying to understand how all of these factors manifest their effects through glial cells, and particularly the microglia and aspects of the brain vasculature. Now, the team that's on board as of today includes a variety of investigators, and they're brought from a range of fields. Now, with me, and I didn't put my face up there because it's not as attractive as Bahara Ajami's, and also because you don't need to see me twice, is an effort to really try to look at the integration of responses from the body by glial cells, which are very responsive to these external signals. Bahara is joining us from Stanford later in the year and is an expert on immune mechanisms. Now, these influences of the body on the brain are mediated in large part, although not solely, through also not just through trafficking cells, as Bahara will study, but also through small molecules and proteins, which are being studied at a population level by Paul Elliott and his team of molecular epidemiologists, and Elaine Holmes, who's driving the analytical chemistries through the National Phenome Centre, but who also shares a particular interest in those species that are derived from the microbiome. I'll describe a couple of them in a moment. Now, we've heard a little bit about sleep, and I think it's Liz Coulthart referred to this. It's something that we think too little about. A third of our lives are actually spent in this funny coma-like state that we call sleep. And what is clear is it's not just a major part of the pathology, but it may hold clues to the way that pathology evolves. Now, Bill Wisden and his colleague Nick Franks are driving work looking at the role, particularly of noradrenergic mechanisms in controlling these phenomena of glymphatic or at least whatever it is that is draining toxins from the brain and amyloid during sleep. And Marco Brancaccio is joining us from the LMB here in Cambridge to look at a most extraordinary area, which I'll describe very briefly in a moment, the role of astroglial clocks in determining cognition. And finally, I just want to touch upon, this is sort of looking at the biology arm. The technology comes in here. Sam Barnes is looking at a circuit level at what homeostasis means in the brain and how it starts failing with aging to enhance susceptibility to neurodegeneration, and then in frank early stages of neurodegeneration, how cells, neurons in particular, respond to these insults in order to maintain function for as long as they can. And then Nir Grossman is working on targeted steerable methods for brain stimulation deep in the brain to modulate not just the microcircuits, but actually also the inflammatory mechanisms and drive sleep. So it's a really, I think it's an exciting program right now. And the imaging overlay comes in most of these areas, have direct imaging components, or certainly use approaches that are amenable to some translation. Now, let me just give you a flavor, and I'll do this really quickly because of time. But Elaine's interested in the gut. Elaine and Paul are interested in interactions between the gut and the brain that are mediated by a variety of metabolites. A particular area of interest that seems to be popping up in strong association with late life neurodegenerative disease are changes in the kyuranine pathway. Tryptophan derived from the gut microbiome is metabolized in the body once it crosses over by enzymes that can drive it down different pathways. Under normal conditions, this is what gives us serotonin, amongst other things. It helps keep us balanced and happy. When the body enters pro-inflammatory states with diabetes or even obesity, enzymes are induced that tend to reduce this flux and drive to kyuranine, which is transported across the blood-brain barrier, interacts with NMDA receptors, and is itself also toxic. Furthermore, she's discovered with Paul that the genes that are associated with Alzheimer's disease, in this case, this is the ABCA7 gene, over large populations, the SNPs for the ABA7 and some other of the susceptibility genes are associated with increased levels of very specific metabolites in the blood that are giving us some clues in these patients to specific metabolic drivers of disease processes. These ceramides are of particular interest because they act as inflammatory mediators directly on the brain. Now, of course, there are other signals trafficking between the blood and the brain, and Bahara has been using Cytov techniques and most recently a method for finer-grained massively multiply parallel assessment of cell phenotype using proteomics to look at cell trafficking into the brains in animal models with a very broad range of neuroinflammatory changes. And what's really important is that I won't go into the details because of time, very specific subpopulations of peripheral blood mononuclear cells are involved in this trafficking. They are distinct in classical inflammatory diseases like experimental allergic encephalomyelitis and in Alzheimer's, but they are found in all. And by identifying these specific subtypes of the peripheral blood mononuclear cells, we can identify, and she started to do this in one case, the specific receptors they interact with on the endothelial cells and factors that may block them. Now, the key to lots of this is about phenotyping. We've heard a great deal about inflammation in the brain. But the challenge of inflammation in the brain in the context of disease is sorting out the yin from the yang. Inflammatory cells adopt a massively broad variety of phenotypes. They can be good players in the sense that we conventionally look at these things, or bad players. They can be neurotrophic and homeostatic or pro-inflammatory. And of course, they can also adopt a variety of states in between. In order to really begin to characterize this, we have to reach into these cells and look at both their transcriptome and, in Bahara's case, the proteome. The transcriptome turns out to be terrifically flexible in this regard. And here, what we've been doing in my group is developing nuclear drop sequencing methods focusing on the nuclei, which allow us to pull down nuclear transcriptomes using microfluidics approaches from which we can sort out in post-mortem human tissue as well as in animal tissue individual cell types, probe on an individual cell basis their transcriptomic phenotypes. Here you see a clustering, one of the early experiments, clustering of cell types into different sorts based on broad characteristics. We can reach into one of these specific pools, such as the astrocyte pool here, and look for the heterogeneity of markers, and then begin to relate these to specific molecular pathways that may be driving the link between genetic susceptibility and lifestyle and environmental influences. And if we know these molecular pathways, in such detail, we can actually begin by similarity mapping to the effects of drugs to map candidate drugs against them. I promised you something on sleep, particularly in this rather soporific time as we enter our early afternoon circadian low. And what Bill Wisden and Nick Franks are interested in is how sleep promotes amyloid clearance, really going into rather as skeptics to test this lymphatic hypothesis and explore alternatives. They want to they have some evidence, both from their group and others, that adrenergic agonists may enhance this, and they're trying to explore the specific cellular mechanisms, identifying those cells, possibly astrocytes that may be primarily responsible for it, and in turn looking at the interaction not just between sleep and amyloid, but at the interaction between sleep deprivation and drivers of neuroinflammation to try to really assess this cycle in more details. Now, they're being joined by Marco Brancaccio later this year, and I think Marco is taking that on with a really elegantly simplified but very sophisticated set of experiments to try to ask a really interesting question, and that is, if sleep is driven by the suprachiasmatic nucleus, which we know it is in most mammalian species, then, and we also know that from recent work, which Marco has been one of the pioneers, that most of the cells in the brain contain their own clocks. What happens when the clock in the suprachiasmatic nucleus and the clocks in individual cells in the brain get out of alignment? So what happens if, the clock at the airport says your plane is leaving at noon, and you arrive at the gate and the watch on your wrist says it's 11:30, whereas the clock at the airport says it's 12:15? Well, you know what happens. You miss your plane. This has happened. But what happens in the brain, Marco has shown, is that if the astrocyte clocks become mistimed relative to the suprachiasmatic nucleus, that in itself is enough, and these clocks in the astrocytes are very, very precise, even on an individual cell basis. If they become mistimed, that in itself is enough to drive cognitive dysfunction. It's absolutely extraordinary. Now, homeostasis is being driven by a number of mechanisms. What Sam Barnes is doing is really looking at the link between structures at the synaptic level and the ways in which synapse density, distribution, and dendritic arborization shift with drivers to altered homeostasis, which seem to be rather fundamental to early genesis of Alzheimer's disease, where excitatory stimuli seem to be relatively uncontrolled in late stages of the disease. He's trying to understand what it is that marks the transition between being able to manage increased excitatory stimulus in the drive towards neurodegeneration and that system failing. And then finally, Neil Grossman is looking at how to modulate all of this, both the inflammatory mechanisms and the neurooscillatory responses. This is a very fancy image, but in fact what he's doing is simple enough that even I understood it. He's shown how you can take a simple two-electrode circuit for transcranial alternating current stimulation, which drives a very broad field here, and take a second one that does the same thing, but drive this one at, say, a very high frequency, like 2 kilohertz, and this one at another high frequency, like 2.01 kilohertz, 10 hertz different. And what he's shown is that neurons don't follow those high frequencies at all, neither do glial cells, but where they intersect, where they beat, like ripples in a pond, the envelope of the power spectrum oscillates at 10 hertz, which neurons and glia do follow, and they actually shift their phenotype. And by adjusting the currents in different circuits, you can position this overlap area anywhere that you wish, given the power that you can inject, and that allows you to deliver deep in the brain. Now that gives you sort of a detailed, perhaps too detailed, flavor of what we're doing. I want to say that the different DRI centers are complementary, but reinforcing in important ways. And I want to just highlight the Edinburgh Centre only because it makes this point very simple to express. They're looking at the neuroglial vascular unit and its interactions with synapses and neurons, linking this to cerebral vascular disease using macroscopic imaging tools such as Joanna described earlier, inflammation and micro and glial phenotypes using methods very much related to those that I've described. And it involves, I think it's really exciting. You've seen some of the output of that from Joanna's talk, work of Joanna as a radiologist with Giles Hardingham, who's someone interested in cell-cell signaling in the central nervous system, stem cell experts, people interested in synapse function, and again, just like our own DRI, complementary expertise being brought together, some of which, like Giles himself and like me, have come from other scientific areas. Now, the latest venture for the DRI, which we'll hear about more soon, is to extend this very fundamental research with an arm that will look at care and technology, trying to push the envelope for technologies in the context of dementia, with a focus very much on discovery, using patients to teach us more about the kinds of things that I've been talking about, while at the same time starting to deliver some real tools to them. Now, a major focus of this, undoubtedly, when whoever takes it on, and this isn't decided, will be at really trying to redefine, or to define in the first instance, what this prodromal stage of Alzheimer's really is. If we're going to treat something 20 years before, what does it look like? What consequences does it have for the person being affected and for their families? We are gathering more and more data about Alzheimer's itself. This is about moving the clock back. And so where does imaging come in all of this? I think there are too many things that could be said, but I'm really very bullish, and particularly so after seeing the kinds of talks that were presented today and what happened yesterday. I'm particularly bullish about imaging. This is just an example. So if we're going to move the clock back, we've got to get more sensitive measures. We've all talked about that. This is work from James Cole when he was at Imperial, using artificial intelligence-based methods, looking at the population variation of brain structural features with aging, showing very strong, an algorithm could be trained on this very precisely, but that if you then go into a specific population, these are the 73-year-olds in the Lothian birth cohort, in fact, people have, brains have a very broad range, very big variance in the relationship of their brain structural features to their chronological age. And if you take a look specifically at these people up here who have brain feature variation that is consistent with an age greater than the chronological age, their 5-year mortality is substantially higher. There are very few ways in which you can predict the future, but this is one. And I think with the massive amount of new data that's emerging in other studies, in this case from UK Biobank, much more can be done. Here, for example, is a refined version of this sort of plot using super healthy ages, and I won't go into more detail about that, but you can use that to refine predictions of disease. UK Biobank has also taught us that using multivariate measures derived from a broad range of imaging modalities in MR, these, for example, are T1, diffusion, resting state, functional connectivity, just to name a few, show extraordinarily strong correlations with a huge range of lifestyle, environment, and I'll show you, and even cognitive phenotypes. These are the negative log P of the associations of these in a mass univariate association analysis that Carla Miller presented with a number of us in a paper last year. And what you're seeing here, this is the Bonferroni correction line. These are negative log Ps of 20, 30, 40, huge effects, and even around cognitive phenotype. There's a lot to play for. In a rather elegant study that Jonathan Marchini and Steve Smith and their younger colleagues published online in bioRxiv, what they showed is if they look, for example, simply at T2 star hyperintense signal across the brain in selected atlas regions, they found extremely strong associations across a number of genes. Here again is the Bonferroni correction line. These are fully corrected SNPs. In fact, what's really interesting is that this SNP here is strongly associated with variation in the in iron deposition in the deep gray matter. It turns out to be the familial hemochromatosis gene, HFE, that's been associated with late life cognitive impairment. And finally, I want to leave you with, there's a lot of blue skies thinking to be done. If we're going to change this disease, we've got to be able to reach in and modulate homeostatic plasticity, to tweak it back, to give it a little bit of help, to tweak it back towards a more normal state in conjunction with other things that we're going to do. I think there's a lot of guidance to be provided by these quite extraordinary experiments that have been done by the EPFL group on spinal cord transection, in which they've shown that if they link pro-regenerative phenomena, drug administration, with activation of neurons in behaviorally relevant patterning, and they did this by epidural stimulation that was timed to gate signals. They found that they could actually make neurons grow in the spinal cord significant distances, find new targets, connect properly, and restore walking to the monkey. And I think the extension of this to the cortex isn't necessarily that far away in using optogenetic methods, which have already been piloted by other groups, as you see here, using very dense optogenetic arrays that allow one to target neocortical changes and repattern them. Now, why do I mention this in an MR context? Well, I had the pleasure last week of being at Leipzig and seeing an extraordinary range of fine mapping techniques looking at patterning of complex cognition in the brain using high field methods from a variety of groups. This is an extraordinary achievement for MR, to be able to actually look at the dynamics of thinking as it's changing on a fine scale. And it's exactly this kind of thing in combination with these non-invasive modulators that I think can bring the kind of transformative technology to Alzheimer's disease. This isn't going to be simple. I don't think there's any easy path, and I'm not sure that these are the right ideas, but this is the sort of thinking that we all should be engaging in. How we can stratify patients, how we can discover new causal mechanisms, and finally, how we can use imaging to actually guide the administration and direction of therapies in much more powerful ways and central ways than we would have thought about before. So let me stop there. Dennis, thank you.
Paul, thank you very much. I thought that was a great way to close, to show how we can use increasingly sophisticated imaging to tell us about how the brain is changing, but actually then look beyond that to say, well, that's fine, but what are we going to do about it? And yeah, it's going to be complicated, but we need to think beyond that and say, so what are the new interventions that we can bring to bear? And I agree, I think it's very, very exciting indeed. Are there any questions for Paul? Bob?
A terrific talk, Paul. Really stimulating. Just one question. Is the DRI going to look into the question of the simplex hypothesis for Alzheimer's disease, which has been pushed by Professor Itzhaki in Manchester for about 20 years or so, but has very recently received a huge endorsement from a major American study showing the very precise co-location of needs to be exactly the kind of area that the DRI should get much more serious.
Yeah, no, Bob, that's a really interesting, that's good to highlight. And I think what you're highlighting is that, if we asked people about 5 years ago whether we were starting to crack, Alzheimer's disease, everyone would nod their head sagely and say, yeah, we've almost got this figured out. And that's what, sent, hundreds and hundreds of millions of pounds onto clinical trials. But we now, I think, are very humble, and we realize we don't even actually know where to start. So I don't know. The short answer is I'm not aware that anyone in the DRI is running with this right now. We're all individual investigators and do what we know what we can think of doing and have expertise in. But I think what you're highlighting is that there's no way for the DRI as it's constituted now to do everything that's important. There are going to be an incredible number of new hypotheses, and some of them may be able to be done effectively within the DRI, and they should be championed if they are. Many of them, if not most of them, won't be able to be done in that context. And the idea of the DRI is not to starve the rest of the scientific environment, but to raise the profile of this disease so that we can begin to get even 3/4 of what cancer gets. To try to catalyze new kinds of studies. So, Bob, it's a great thing to, we've got to chase this now. Someone needs to.
Someone's got to,
Yeah.
Do we have any other questions? Frank?
There's been kind of an ongoing interest between thoughts and brain in terms of depression. There kind of indication of overlapping mechanisms.
Yeah, so I think we're, so the first part of the answer is that I think we're in a very primitive stage still. So when we talk about the gut and the brain, there are a few things that we actually understand quite well in very peculiar mice that are raised, without any microbes, such as, a massive role for specific species in driving the development of tight junction proteins in endothelial cells, and that's crystal clear. But that's in a very peculiar model. And we know that some of these metabolites derived are CNS active, and obviously serotonin, the links between serotonin and depression and psychoses are well described. So that has been, areas like those have been pursued very much. I think I'm very interested in the comorbidity between affective state and dementia. As you know, depression is one of the major comorbidities of late life sporadic dementia. And that really makes it tempting to think that they are mechanistically linked. Whether the gut is the driver of this or contributes to the susceptibility or helps to drive it, I think is something we've got to discover. And it will be just by the convergence of thinking from what we would call traditional psychiatry and this kind of science that we're going to find the answer. So that's a long way of saying I don't know, Franklin, but I think it's something we've got to keep our eyes open for.
Questions? Okay. Well, in that case, Paul, once again,
Thanks.
thank you very much.